Great. Yeah. So we thanks everyone for wonderful talks, and we do. I'll join you over there in a moment. Maybe I'll just do that right now. Yeah, we did have some questions going, but let's start with the question you had, Stu. You want me to just pass it over to you? Yeah.
This is from very, very long ago, um, like 60 years ago. So here was the question then. It was known that late replicating regions were often heterochromatic. It was also known that different regions replicated at different times in an organism. And so I wondered at the time, could it be that replicons activate one another in some pattern that's then associated with late replicating guys becoming inactive as part of the control of cell differentiation? So a long time ago, we did an experiment in Physarum showing that if you fuse early and late replicating stuff, late replicating replicons replicate early when exposed to early S cytoplasm. So I thought, gee, maybe, and then I went off to other things. About 2 years ago, I happened upon some article that seemed supportive of that general idea. The question being, is there a relationship between the order of replication and suppressing gene activities, and so cell differentiation? So do you know anything about that? Is there work that supports that, or is it just wrong?
These polycomb targets that I talked about, interestingly enough, they are often at the boundaries between the late replicating and early replicating regions. Not all of them, but typically. And in Drosophila, there's been some very nice work showing that some of these polycomb targets are in late replicating heterochromatic regions, and then when they need to be turned on, they actually move into the interior and then back again. And so, yes. So, yeah, and I don't think that's been demonstrated yet in mammalian cells, but it's been shown in Drosophila. I think one of the reasons for sort of an in-cis relationship is these are megabase regions, obviously, and there's all these loops, these huge loops that go to the nuclear periphery and then back in again. And the chromatin of these megabase regions are often coordinated or similar. And so some of the sort of coordinated behavior is likely in part attributable to the chromatin marks, the histone modifications, et cetera. That's about the limit of my knowledge. I'm a DNA methylation person, not a replication person.
Okay, well, we thought we would just start off with this question. It was asked in the first session, and I think you guys have done a good job of touching on it. But the question being kind of from your interactions with Ilya, kind of what you took from those interactions that kind of added to your research or made you excited or kind of hopeful in your research path.
Yeah, thanks, Dave, for that question. And fortunate for us, we had a lot of time to think about that one. I think the one thing that struck me was that there's not any one interaction or conversation, but a prevailing set of memories of how all the interactions were always positive, and that anytime you did interact with Ilya and you had a conversation, you left that interaction feeling positive, feeling inspired, and feeling excited to keep working. I think that he was always encouraging us to be excited, but also curious, and also to have rigor in our work. I think one motto he always had was like, do good work. It doesn't matter what else is going on. Things, there could be problems, there could be trouble, lots of things could be happening, but at the end of the day, is it good work? Have you brought rigor to the question? at hand? Is it an important question? Have you asked it appropriately? Is it interesting? Is it an important question? Like those kinds of conversations were always very important to me and very insightful, and I always appreciated them.
Thank you, Nyasha. I'm not sure what to add above everything that has been sort of said already today. My initial years with Ilya, I thought, wow, he's somewhat sort of loosey-goosey as a manager. And as you already said, you don't work for Ilya, you work with him. And so in the beginning, I thought, in a way, not very highly of that. But sort of with the years and sort of in a retrospective way, this was a way in which he was very empowering to the people in the lab. Indeed, one of his main mottos was do good work, right? And so he very much encouraged us to do that, thinking outside of the box. The work that Peter showed on multi-skill representations coming from a completely different domain from me, and was like, yo, this may also be the case in biology, right? And we started to apply it, and it actually led to very interesting findings. So the magical way in which he empowered everybody in the lab has been, really remarkable. I don't know what the recipe is in which I'm able to do that, but I hope in one way or the other, I'm also being able to channel that particular skill.
Yeah, what I would add is, and it's been mentioned today before as well, is that when you talk to Ilya, he displays just this incredible curiosity. He just wants to learn and understand. And the other thing that has struck me with all my conversations with Ilya is when you talk to him, he gives you his full attention and you feel valued and that he's listening to you. And I think that's why so many people here feel that they're very close, or we're very close to Ilya, is because he gives you that feeling of you're important, I'm listening to you, I'm looking at you. And that is something that you don't encounter that often.
Can you remember any of his Russian jokes? I can remember one. Okay, this one is not so much a great joke, but he did tell it multiple times. He would say, the difference between American professors and Russian professors is in America, they say, your student brings you some work and you go, oh, wow, you did so much great stuff. This is so cool. Great job. But there's a little problem here. And in Russia, the student brings you some work and you say, oh, this is terrible. Oh my gosh, I can't believe you're my student. This is abominable. But maybe there's something good here. And so that was.
Dave, you do a very good job of joke telling, and so did Ilya. And Ilya told me lots of jokes, lots of dark Russian jokes, and
There's a lot of
laughed
dark
out
ones.
loud a lot. I know for myself that I'm actually very bad at remembering jokes and also very bad at retelling them. So I'm definitely not going to try that. I will say that for our lab, for the Ilya lab, it's always sort of fantastic to be sitting behind your desk and then hearing Ilya with somebody in his office laughing out loud, right? This always brought a big smile to my face. So, I think that, the humor aspect and being able, in order to take something serious, that you also have to be able to make a joke about that, I think was also very central to the way in which he looked at things. Any jokes from you, Nyasha?
I definitely remember his Russian gangster imitation, that he would imitate a Russian gangster once in a while if you said the wrong things. I was curious about one thing about from you who have worked as part or were part of the TCGA in such a deep way, and that project, the way as was described earlier, was very kind of deep dive with multiple platforms. Yeah, with multiple platforms, but looking at bulk tumor data. And in the past years, as was also pointed out, and has been said many times, we have many other platforms at our disposal, and it's talked about how much they shed light on things, single cell sequencing and ATAC-seq and epigenomics, as well as spatial methods. I was curious, can you think of any specific examples from TCGA findings, or I was wondering about specifics where we learned something or were puzzled about something where years later, when these advancements have been made in assays, are there specific examples where it's like, aha, now I understand what was going on, or now I see, like, why we couldn't figure something out? And can you think of any like specific examples that way? I think Peter, you kind of led into that in the last part of your
Yeah, I talked about
discussion. Yeah.
GI today, but one of the things that we found early on, very early on, was when we did unsupervised clustering with DNA methylation of glioblastoma, we found a small cluster that had extreme hypermethylation, sort of like the EBV SIMP in gastric cancer. And we found, interestingly enough, that it was extremely tightly associated with mutation in IDH1 in the samples that had been sequenced. So we talked to Li Ding, and she had only done about half of them or so. So she pushed to sequence the rest for IDH1 specifically. This was 2009, so very early in the project. And so we got all the sequences, and then we ended up publishing it in 2010 in Cancer Cell, a month after the Verhaak paper that we showed, I think, on expression. No, that was ovarian, but on the expression subtypes. And so we didn't understand why IDH1 was causing this hypermethylation phenotype, but a few years or not too long after that, it was revealed that that IDH1 mutation creates a novel oncometabolite which inhibits both demethylases, both histone demethylases and TET enzymes, which start the process of DNA demethylation. And so then it became clear. And so that was an example. And the EBV SIMP, again, in gastric cancer, we found this, we reported it. My lab didn't have the bandwidth to sort of pursue that mechanistically, but then there have been lots of papers since then that have dove into the mechanism. So I think one of the values of TCGA is hypothesis generating, right? We made all these discoveries and associations, but the people in TCGA didn't always have the bandwidth to pursue them. But there have been literally thousands and thousands of papers citing the TCGA data and reports that have done exactly that. So I think it's been hugely valuable.
Yeah, I think Peter's examples are very striking. The one I also thought was from glioblastoma in particular was initially people thought there were 4 subtypes, molecular subtypes, and molecular subtypes based on glioblastoma, based on gene expression profiling primarily. And over time, I think it emerged that it's probably 3, and it was bulk RNA-seq, and perhaps one of those subtypes was being driven by normal cells that, because it was bulk data, were in the mixture. And so I think what I've come to appreciate over time is that I don't think I fully grasped tumor heterogeneity as a concept prior to TCGA. And so I think if there's anything that I think about a lot more now is we did find, TCGA did describe a lot of interesting things, and we did make a lot of discoveries, but I think that there are some things that are maybe obfuscated because of heterogeneity, and some things yet to be discovered that we do need these newer single cell technologies for.
No specific examples from me, just a general observation that in TCGA, but in many other projects where we unleashing all these novel measurement technologies, the patterns are just jumping at us, right? This is to the point that Lee made in terms of generating good data. If you generate a whole bunch of good data, especially when you have many domain experts that are participating in that, then you're going to identify many novel patterns, and things that were first mysteries are becoming clear. And we will also see this with the single cell revolution as we start measuring many things with single cells. Certainly not necessarily the case that we need complicated AI methodologies in order to find these patterns. I think it is more the matter of measuring good data with novel measurement technologies.
So, go ahead, actually. Do you want to ask a question?
I think a really interesting question for the whole cancer field is, what are the transformational technologies supported by AI approaches that are going to really take us to the next level? For example, single cell analysis opened up enormous dimensions of new opportunities and things like that. And certainly the thing I was involved with, automating DNA sequencing, has really, so what for cancer research, and spatial biology is really fascinating, but is there anything next? Where are you going, are the field going to go?
Well, part of it where AI may be a big help, and I'm speaking a little bit outside my field here, but might be in not so much the novel technologies, but the integration of all of this, right? So, and I think we'll be limited by the amount of data with associated phenotypes and clinical outcomes. As we get more and more of that and the value of those relationships become apparent, the data will become big enough and complex and high dimensional enough that perhaps AI can really accelerate identifying novel discoveries there that if you look at how we did it in TCGA, that was very painstaking and manual with a lot of back and forth and curation, et cetera. And AI should be able to do that far, far more efficiently by probably orders of magnitude.
But to go with that, I think what you need is to follow the evolution of the tumor growth, because dynamics really give you the capacity for deciphering a lot of complexity that 2 points don't. So I think that following the dynamics of disease is really going to be important.
Yep, I would agree with that. And going also along with the line in which Sui often thinks and talks about cancer, I think it's going to be very important to nip it in the bud, to be able to detect it as early as possible, because the longer you have this oncogenetic process, the more difficult the treatment ultimately is going to be. And I think that there are lots of opportunities there.
That's what I was getting at also with these polycomb target hypermethylation occurring in normal cells. If we have the ability to screen for that and field effects, et cetera, as part of risk screening, that may clue us into the risk of developing cancer as we see what has already undergone these epigenetic alterations, and it's not just mutations as well.
So Peter,
that's a good question, and it could be stochastic. It could be there's insufficiency in some cells of polycomb components. One of the experiments we're planning to do is deplete polycomb and see if that allows methylation to come in more readily, for example, because there's this competition for repression. We have the heterochromatin H3K9 trimethylation, we have polycomb, the K27 trimethylation, DNA methylation, and then transcription ties in with that too. And they're all sort of competing forces. And a lot of experimental systems have shown that when you remove one or add one, that you perturb the others as well. And so where things go in the genome are affected by the relative amounts of various marks throughout the genome.
so you may be answering what I want inarticulately to ask. So how does one get from, in particular, Peter, what you're talking about, to causal roles, quotes in cancer is a very broad statement. For example, I'm used to thinking about altered patterns of gene activity and cell types and so on. How do you think causally about the roles of what you're looking at in terms of what a cancer does?
Yeah, we, in terms of causality, we tend to use manipulations and mouse models, for example, to show the reason I'm working on DNA method. I'll tell you a story. When I was a postdoc, I hated DNA method, or as a grad student, I said it literally to someone at a conference. It was so boring. I said, if there's one thing I'm never going to work on, it's DNA methylation. And then I went to Rudolf Janish's lab, and Lee in the lab had made a knockout of DNMT1. And then I realized, here's a tool that we can use to manipulate the system. So instead of taking a human tumor and look at the DNA methylation changes, we can take a mouse model, change the DNA methylation, and see what happens to the tumors. And so we crossed the DNA methyltransferase knockout with the APC min mouse, which develops intestinal polyps, and we essentially could completely eradicate polyp development by reducing DNA methyltransferase expression, which is needed for the CpG island hypermethylation. So that suggested that having enough DNA methyltransferase activity was just as important for developing polyps in that genetically predisposed model as the APC mutation itself. So that's an example of where we found evidence for an important role in DNA methylation. And that's why I've been working on it now for 30 years.
Oh, I was trying to remember. Oh, your question, Lee, about which technologies are enabling and how can AI contribute? Where do we see the next wave in cancer, and where's the field going? It's a difficult question to answer, but I've been thinking a lot about how I think there's a tendency for us in the field to think about one size fits all, and also to hit like a home run right out the gate. And there's this idea floating around that there is some degree of incremental changes or changes at every step in what we describe as the cancer continuum, prevention, early diagnosis, treatment, and then surveillance or survivorship, right? So in the prevention space, like identifying people who are at high risk and using AI modeling to like for example, in the work that you were doing at Aravail, Phenome Health, who is likely to be at high risk? Can we identify them early and have lifestyle and behavioral intervention so they never ever develop cancer? So having AI in that space is a feasible option because we know how to build those type of models, using clinical EHR data. The diagnostic space, I would say, is in the lead because it's imaging data, and AI and images are just optimally placed or are in the lead in the way to work to be optimally placed to make a choice. And right now, I had been working on a presentation for something, and there's 527 or so FDA approved deep learning models in clinic right now for pathology assisted interventions in some way. So I think in that space, sort of diagnosis, radio oncology space, AI is already in place. But I don't think there's any one size fits all across the entire cancer continuum. Yeah.
Okay, this is just a fun question, but if you were the head of the NIH and you were able to just award yourself,
You might be rephrasing my question. Yeah, it was kind of generally,
generally,
not just for yourself, but for the
Not for yourself.
community.
for the research community.
Yes,
But
for
what kind of, if you could design an atlas, like a HTAN, Human Tumor Atlas, kind of a large consortium direction where you were going to fund labs for, a particular avenue, what would you do?
I actually told an NIH officer this last week. I was like, we just need to do TCGA, but with like the reflection of the people who get the diseases.
Yeah, great answer. I think, yeah, I think we were kind of speculating like, so NCI is always, I mean, was inspired by the TCGA program. It was considered successful, and there have been different, that's branched out to different, different other, as a model for other kind of Atlas efforts and kind of, so yeah, the general question, what, like, just to follow up on what Dave was asking, like, if you were in charge, what kind of an Atlas or cancer genomics program would you put together? And Nyasha gave the first answer. You know, it would be to be more inclusive of the population. And are there other things where what would you do if you were in charge?
Do I have an unlimited budget or? Well, if you recall, those who were at TCGA in the very beginning, the biggest challenge was sample acquisition. And 95% of the samples were rejected because of tumor purity, lacking clinical information, whatever. And I'm involved now with some projects where we're trying to compare primary tumors with metastases. And the challenge is always you can get fresh frozen metastases of a patient who's recurred, but then identifying the primary with, for example, breast cancer might have been a decade earlier, so an FFPE block somewhere that has been oxidizing. So it's a very long-term project, but if NIH invested in better sample acquisition and annotation prospectively of a variety of cancers and storing them in ways that are compatible with many of the modern technologies, over time, patients will recur. You can look at metastases, you can look at progression and things like that to the dynamics that we've been talking about, because that's missing, understanding sort of the pathohistory of the disease history, and that would tie in very well then if you applied the spatial tools to these samples to understand how different subclones may progress, et cetera, in a more detailed way.
Actually, there's great population because the individuals that have survived the first cancer in the future constitute at a given time 20% of the new cancers that come up that year. So if you want a great population to look at, that would be the one.
Is that with chemo or radiation or all of the above? I mean, what?
It's all of the above.
So let me elaborate on my earlier comment. So it was a little short, but I think that we know we do also have silos in cancer, right? We have epidemiology, we have public health, we have social scientists, we have genomics persons, we have molecular biologists, and we're all doing our own thing, and we don't talk to each other necessarily. But epidemiologists know a lot about what causes cancer, what are the risk factors, what's associated. But we don't, they have cohorts that they follow longitudinally, but they don't collect biospecimens. So they have cohorts with the detailed phenotypic metadata carefully collected and annotated for 20, 30 longitudinal years, you know, but then they didn't collect biospecimens. And then though they'll have germline DNA and they don't have the tumor samples. So something is not right here somewhere. For example, my group started working with all of us data to try and assess the feasibility of looking at cancer risk, building cancer risk models that combine germline genetic variation and clinical data. There's also participant provided survey data, lifestyle data, Fitbit data, sleep data. So there's a wealth of data out there, but it's never collected in the same individuals over time. And then you have their baseline data as well as their cancer data and what happens after. So I think the real investment is to collect the right types of data in the right persons over the right timescales. So we can even begin to tease apart all the questions we have about what's really driving cancer, both from a genetic and molecular perspective, but also environmental exposures that are contributing to cancer.
Instead of collecting data that you don't have already, take all the data you do have and invest in algorithms that can actually anticipate patient trajectories through those periods, which I know some people are doing that for infectious diseases, and it's actually pretty effective. But they're novel algorithms, and you need a ton of data to do it. But there is a ton of data, as you point out.
I will talk with you after, Jim. No, so we're doing some of that. There is, but there's always the question of, well, what's going on with the tumor? They never have, if they have the clinical data and the sort of lifestyle data, they might not have tumor-specific data. Or if they have tumor-specific data, then they don't have the clinical variables or the lifestyle other epi variables. So either you get the normal related data or the tumor related data, but never the two shall meet for some reason.
Let me also give this a try then. But in thinking about this, I am very heavily influenced by Ilya and by Lee, and I think that big projects is the way to go, right? So if one, has a whole bag of money, then that is something that you could do. Stu was talking about the notion that many people that are now working in oncology know very little about dynamical systems, for example, right? And so these hospital organizations are historical institutes would have a huge momentum, and that's very difficult to sort of, to break that up. Lee has often talked about starting a new hospital of sorts, right? And so I would very much agree with that vision. I think it has been the very large projects over the last couple of decades that have led to the breakthroughs. And so I think that is one way in which one could approach it if you have a very large bag of money.
So I think another way to do population studies that would be very powerful is to go to a place like Finland, which has absolutely superb biobanks. And often they'll look at normal people for 30 years. And what I'm saying is you could go to the biobank and you could select a cohort that had X that you were really interested in. You could accumulate 30 years of knowledge, and then you could enroll them to look dynamically at what's happened in 32 and 33 and 34 and 35. I think that is going to be an incredibly powerful approach to getting at this whole question of dynamics and being able to look across the history of the initiation and progression of diseases, or even just to follow ordinary people and look at aging. So I think thinking about unusual ways to capture and save time on these populations like that is a really important direction.
Are we open for questions? I guess, sorry,
Yes,
I
please.
have a
Yeah.
general question to all of you concerning Ilias or in TCGA, and something I've always wondered, and you might have the answer. This is schism here. We noticed that. We saw this morning, and Stu talking about dynamical systems. People don't understand it. So this is engineering rigorous formal approach that seeks the mathematical description of prediction analytically and emergent properties. And Ilya knew about this. But then there's TCGA, which essentially is a reductionism on steroids, right? Nothing against reductionism, but it's collecting data and hoping to find something. And Ilya could seamlessly move between these two, but they're two totally different philosophies. And I wonder
how you felt about that, whether you were sometimes confronted by doing some modeling as opposed to data analysis, and so especially those are two both, even Theo.
Ultimately, I think it is very attractive to think about things as a dynamical system and also ultimately able to model them. At the same time, I think that sort of throughout biology, we lack the data to actually learn the parameters of these dynamical models such that we know how we can perturb, the system, drive it back to a healthy state. So, the notion that one needs a very large scale, very high quality dynamical data in order to start learning these models, I think is the case. The TCGA provided perhaps just a snapshot in the lives of cancer patients at the bulk level of grinding up the whole tumor and seeing what is there. And so, this is ultimately not the data that we want. And at the same time, it is definitely a very good step in that direction.
To all of you, I'm hearing this for the first time. I can almost say what I want to say. Forgive me if I screw it up. There's indefinitely many facts one can collect about anything. And so the question, a almost formulated question is, on what grounds does one say this set of facts is important? A classical case is that in physics, it turned out position and momentum was important. And if you want to know something about the swinging of a pendulum, the color of the pendulum doesn't matter. So figuring out what's relevant is really hard. And a case in point is Jacob and Monod answers the question, how could you have the same set of genes, but different genes are active in different cell types by showing that they turn one another on and off? It's a poorly formulated question, but on what ground with respect to something as vast as cancer, does one say, I have grounds to suppose that this is relevant and worth looking into in such and such an expense, given other things that I might be doing? I know this isn't a well-formulated question, but do we have a way of thinking about it? I'm giving it back to somebody. I have no idea what the answer is. I think so.
That is a problem, and that's, I think, best expressed in Darwin's quote that, without a theory, one might as well go into the gravel pit and describe all the pebbles in all its colors and shapes, right?
Yeah.
And not know what you are doing. I wonder what the part of TCGA is that. We have to
What?
be, I wonder what the part of the TCGA is that. And we have to be very honest about ourselves, right?
But this gives me a chance to tell you all something. This is a wonderful moment in my life. I was doing fruit fly genetics. Gerald Schubiger was a fruit fly geneticist from Zurich. And he said, the trouble with you, Kaufman, you got the theory before you got the data. And I loved his accent. But also it's true, you have no idea what the relevant data is until you've got some theory. And it's always true. I mean, Gerald was right and I was right. So how do we judge? I know it's a useless question.
I don't think it's a useless question, and there are a few ways you can answer it. One is obviously clinical impact, right? So some of the subtypes that have been discovered in TCGA have been shown to, for example, respond differentially or different prognoses, et cetera. And clinicians are using these to inform clinical trials and things like that. That'll happen even more as we learn more about the molecular features.
One answer to my question, you look at something, it turns out it's relevant.
Yes.
So you didn't know beforehand, but you got to go ahead anyway.
Yeah.
Mike's always had to do that.
And
we always have to find out something that might work and we didn't know what it might be.
yeah. And Theo mentioned that we're lacking a lot of data in biology because there are so many variables. And so what TCGA started to do was just systematically collecting data. And then one of the things that you have no idea what's going to be important, right?
Yeah.
But one of the things you can then find is not just clinical impact, but also associations between different types of data that you didn't know about or didn't recognize beforehand. I gave an example of this DNA methylation phenotype in glioblastoma linked to IDH1 mutation. Nobody knew about that. Nobody had that mechanism was not invoked or anything like that. And that actually led to the development of IDH1 mutant inhibitors that are in clinical trial, et cetera. And so I think that those kinds of horizontal associations can lead to new insights in mechanisms, disease ontology, and with clinical impact.
I think the other point that's important is serendipity. Serendipity lets you look at something you have no idea about and explore who it might be related and how it might be related and things like that. So I think we don't want to get too wrapped up in saying we have to do this because we'd like to translate it to the clinics. I think it's key to explore new dimensions of data space. I think that's really a big driving force for technology and biology.
I want to make one other example, and that is before TCGA, we had this list of oncogenes and tumor suppressor genes, and that's what we went into thinking that we would discover more of. A whole class of mutated genes that turned out to be very important turned out to be epigenetic modifiers and regulators, ARID1A, et cetera. It was on no one's radar screen. It wasn't picked up in any of the model systems. So that's a discovery that came out of all of these large-scale cancer genome projects that was unpredictable. And that's why you first have to collect the data, collect the postage stamps before you can start to do stuff with them.
First of all, forgive me if I'm not very articulate, but I'm just, starstruck by all of these people here. I just wanted to offer maybe a naive-ish view on the question. One, I know you've read Rosen because I read your book, and yeah. So it's a theoretical biologist, I think from the 80s, 90s maybe. He pondered a lot on complex systems and organisms as complex systems. And I like one of his students' description of what a complex system is, and that is a system such that no single finite description of that system is enough to account for all of our interactions with it. So that's to your point of which set of variables. I don't think for complex systems, I think that that's the property, is that there's never going to be one set of variables. It's always going to depend on the problem at hand. And then sort of related to that, it feels like you've already all given the answer to this, but it feels like we're all never going to run out of jobs because we're always going to be able to theorize with a subset of the variables at hand, guide the next set of variables to measure. And so the experimentalists also still have a job. And so there's this other concept that I think is interesting in the context of complex systems and relates to your presentation as well, that of computational irreducibility or irreducible complexity, in the sense that there seems to be just certain processes that we just need to wait out and see. There's no kind of equation that we can write that tells us the answer of this process before it happens. And in a poetic way, maybe I'm being too much of a romantic here. It might be that the scientific method in this case is irreducibly complex, and we just need to keep iterating in the sense of coming up with new variables, coming up with new experiments, and we happily build the train tracks in front of the train and continue like that.
Thank you for the great comments, and it's always good to have the audience feedback and questions from the audience as well. And thank you as well to the panel for great discussions. I hope we didn't put you too much on the spot with difficult questions. I think I'll just take the opportunity to wrap up and thank everyone. When we were thinking about this early on, this idea of having a scientific conference, one of the questions came up as to how should we title it? And we did end up, we through a combination of discussions calling it Resonance, and that was both for, that seemed to match very well, both in terms of the musical connection to Ilya, but also it seems so well demonstrated today with all the presentations that we heard, both in terms of how Ilya's work impacted multiple different fields, and those fields individually, those being amplified through these connections, through this idea of resonance, and both in terms of the scientific work, but also the interactions among the different people involved, who are, we're blessed to have people here who interacted with Ilya through those years and describe that so well. I'd like to thank our sponsors for this symposium, GDIT, and for everyone. Thank you to everyone who attended, including Ilya's family who is here. We're so fortunate to have you here as well today. Thank you to all the speakers, and thank you to our ISB staff who got this together, both in terms of getting the idea off the ground, and the implementation from our facilities and operation folks, a team led by Shana Braga. Dominic Lewis played a big part in getting this all together. Allison Kudla, I don't know if you're still here, helped get the wonderful the web page and that beautiful image, that drawing. She organized and put all of that together. So thank you to Allison as well. And yeah, thank you also to those who attended remotely and couldn't be here for attending. With that, we'd like to wrap up the day. It's been an amazing day to hear all these different aspects of the scientific work that Ilya influenced, as well as the personal level too. Yeah, yes, and that's, yeah. Thank you all for being here. Yeah.